Texas.
Yeah, yeah.
So welcome to the regular monthly meeting of the Fort Worth chapter of the Native Prairies Association. And I'm Suzanne Tuttle, and I'm the treasurer for the chapter. And Kate Morgan, our president, is here. Hey, Kate. And Michelle, our secretary, and Lorraine, our vice president. So all of our officers are here, and lots of other members and lots of guests from across the state. And we really want to welcome you to our meeting. So our speaker this evening is Dr. Bruce Benz. He's a biology professor at Texas Wesleyan University in Fort Worth. He has a really interesting career and did his graduate work on different varieties of heritage corn in Mexico. So that's a whole nother very interesting topic that I've heard him speak about. But tonight he's going to talk about one of my favorite endemic plants, Silphium albiflorum, the white rosin weed. And Texas Wesleyan University, for those who are not from the Fort Worth area, it's a private college very close to Tandy Hills Natural Area in East Fort Worth. And Bruce has adopted Tandy Hills as one of his close-in research sites for his students. So he's been doing this study on the Silphium albiflorum and their pollination strategies in isolated colonies for quite a while. And I'm going to let Bruce take over. And Bruce would like for y'all to just interject questions anytime you might want to. Don't wait till the end.
Stop me when the urge comes over you. So thank you, Suzanne. And I'm happy to be here. I know most of you, Michelle and I have, and Suzanne have spent hours together at Tandy Hills. Not the last time was 2016 when we ran the Bio Blitz out there. It's a great place, a remnant prairie. It needs people interested in it. One of the species that is fairly common in Tandy Hills is white rosinweed. And white rosinweed is I think a fairly typical cross timbers ecoregion endemic, here shown with another cross timbers, not endemic, but largely restricted, the scissortail flycatcher. And what I want to do—
Could I interrupt you just a moment? Suzanne, did you want to record this? I can't push the button because I'm not a co-host.
It's already been recording. I think it's set up to automatically start when the first person arrives. So it's been recording.
Okay, sorry.
Yeah, I checked it when I first got here, so.
Okay.
Okay.
Sorry.
So what I want to talk about is a little bit about the species in general, what we know from the general lines of inquiry that exist from citizen science, and then where that line of reasoning took us to actually ask more specific questions about the species and its numerous populations, leading us through the demographic approach, then followed by the genetic approach, and then more recently referring to sort of a community ecology approach that I had not anticipated, except when I first started, that has become sort of a real avenue of intriguing study that doesn't seem to have any end. And at that point, I'll sort of surrender the microphone back to whoever wants to ask questions. So when I started working in Tandy Hills in 1997, I recognized Silphium albiflorum as a relative of Silphium laciniatum, the compass plant. As a grad student at the University of Wisconsin, I spent time collecting for some of the systematic botany courses, and we would visit all the protected areas in southern and middle Wisconsin. And Silphium laciniatum was a common organism, and it is it merits discussion and interest because it is such a striking plant. And so when I encountered albiflorum after moving to Texas from Mexico, I thought it was at first, until I moved slightly down slope in Tandy Hills from the top up on View Street, going north down to what I call the Trinity River Breaks, and encountering Silphium laciniatum. And so I could see in one place this relationship where they seem to have distinct patterns of occupancy, with the laciniatum growing further down slope, whereas the white rosinweed much more common up on top, where the limestone crops out. As I said, Silphium laciniatum is up here in Madison, Wisconsin, and areas nearby. The genus is well known. It's endemic to the eastern United States. So from an evolutionary point of view, the whole group really is worthy of study, because the various theories about how adaptive radiation and speciation and what we know as the taxon cycle appears to actually be fairly abundantly demonstrated by the various narrow endemics that have evolved out of the wealth of species and geographic opportunities in the eastern United States. So this is a map from of the species that were of the locations for Silphium albiflorum in north central Texas. And we can see here the Metroplex, Highway 35, west and east. So Fort Worth, Dallas, Waco down here, getting down to Troy, and further south down here to Austin. And so this is the aerial extent of the species. And it seems to prefer these light blue dolostone carbonate sediment that's greater than 50% by weight of dolomite, or the light green areas, the sedimentary particles of sand and consolidated sedimentary sand, and less so the yellow, the glacial marine sediment that were eroded and derived sediments from a marine environment. And the thing about Silphium is that if you can see the dolomite, the white limestone, in the aerial view of Google, of iNaturalist, without doubt, you'll find Silphium albiflorum. And it's a very competent plant when it comes to living in these limestone outcrops, suggesting, in fact, that it is able to cope with the relatively high pHs that are associated with the limestone environment. So when I started working on Silphium, I talked with Dr. Carey, who is an old colleague of mine from southwestern Colorado, who is a palynologist, a student of the botanists at UT, and she was working for the Texas DOT, and of course clearance for road building and for power lines and pipelines and the like required an EIS statement that involved looking at the impact on plants and animals. And I asked her about, Silphium albiflorum. I said, it's endemic and it's narrowly endemic. So I suspect that by its very nature, its rarity would suggest that it's probably on the verge of threat if humans are encroaching, or if humans are not encroaching, it is probably a species that is sort of at the end of the taxon cycle and maybe not long for this world. And I was told by Dr. Carey, she said, no, it's not even on our list. The species appears to be, thriving in Texas and doesn't need protection at this point. So I looked at the IUCN criteria for evaluating extinction threat. And this is the basic categorization, right? That not evaluated was where silphium was when I started. And it was probably not evaluated, and we don't care actually when I started. And it was in fact data deficient. We had maps from herbarium records, and there's a couple of herbarium records that are in the global environmental facility that is a clearinghouse for the iNaturalist data and other herbarium records, and it shows that Silphium albiflorum actually is known to occur in the panhandle of Texas as well as in southern Nevada. Well, so I've looked at those specimens. I've looked at pictures of those specimens, and they're not Silphium albiflorum. They're not even probably Asteraceae. In any event, it was obvious that the deficiency of data was enough that it was worthwhile engaging my students. And I have to tell you that my students were not particularly interested at the outset. In some cases, I took them to Tandy Hills or to Stella Row and Prairie, kicking and screaming because they didn't want to spend time outdoors in the middle of September or October in Texas. In any event, what we decided to do, that in order to actually assess extinction threat, we needed to actually embrace the IUCN Red List criteria. We had to look at geography. We had to look at population structure, both age and vital statistics, as well as genetic structure, because it's associated with demography, and then community relations. And that's what I'll talk about in the course of tonight's talk. So the IUCN criteria that is used to quantify the level of threat in a very general way incorporate two measures of geographic distribution: extent of occurrence, the total area the species occupies in square kilometers, and the area of occupancy, the area in which at least one individual is present within a 2 kilometer square area. This is standard procedure for constructing conservation research, and although there's been a lot of discussion about whether or not EOO or AOO or both or some modification of them is appropriate, these are still relatively simple means of assessing threat, and are generally used and embraced by the IUCN and other conservation researchers. So what we did when we started was, and this was after iNaturalist had begun to record evidence for natural biota. And so what we did is we looked at the AOO and the EOO of silphium. And according to the evidence today, the extent of occurrence, EOO, is large enough that it, and in fact, does not have its least concern. But if we look at actual area of occupancy, the amount of area encompassed by the individual populations, it is in fact endangered. And I have to tell you that my students got a kick out of this, because when we started to do this in earnest, there were very few recordings of silphium, of white rosinweed, in Texas. And so in 2015, it was low concern, but it was endangered. In 2016, as more people recorded this beautiful white aster that is in full bloom on the 4th of July, was still endangered. And as we moved up until the recent, there has been some suggestion that in fact, we may get to a point where the areas here, if they in fact contain Silphium albiflorum, might in fact reduce the vulnerability. Now, I say that in the same breath, I want you to realize and recognize that the area that is of the highest density here, and it's recorded in the Metroplex, so you can expect people are quick to pull out their phones, whereas down here south of Brownwood or over here in Comanche, it probably isn't as probable that people are going to whip out their phones and take a picture of white rosinweed. So maybe this is an artifact of population and the number of individuals with cameras in the vicinity of the species. And so this is a population in the right of way on Highway 67 north of Midlothian, which is south of Dallas. And many of the populations observed in iNaturalist and collected by herbarium botanists are found in the highway right-of-ways. And some of my colleagues actually poo-poo the evidence as being an artifact of road building. And yet, if you actually look at these populations, there's a population south of Temple, for example, where the individuals are actually coming through the asphalt, and which suggests, in fact, that they were paved over and have come back. So they predate the construction of some of these roads. And the outcrops that are formed by road building are the likely, places where silphium seems to thrive. So maybe we're, by building roads, actually enhancing its territory. Like so many things in life, the road building has a downside as well, and that downside is that mowing contracts let by counties seem to focus, at least in some parts of the range of Silphium albiflorum, at about the time when the plant is in the midst of flowering. And the result, of course, is that the plant does not produce seed. It is a perennial, it comes back from rootstocks year after year. But we don't see then any sort of maintenance of genetic variability because it's not reproducing sexually. So I said the concern for population viability is fundamental to the IUCN Red List criteria. Population reduction based on direct observation, or the projected inferred based on effects of introduced taxa or parasites. So what we did is we sought out to actually characterize populations in a manner that is commensurate with the way it grows. And so we see here, this is a data recording sheet on which a number of individuals of Silphium have been laid to take a photograph. And what we see is that the plant produces about a leaf a year, maybe slightly more, maybe slightly less, depending on the environment. And although this has been only marginally tested, it does seem to suggest that the greater the number of leaves produced, the older the individual. And so even if we were to not actually identify the number of leaves as an indicator of age, perhaps in years, we could use the 2 here, the 4 here, and the 2 here as suggestive of relative age. And so what we did is we use leaf number as a way of describing population structure. Now, we also then, in usually the fall of the year, count the number of seeds produced by an individual by actually pawing through all of the capitula that are produced by every individual plant. And what this allows us to do then is to actually calculate and characterize using what are called static life tables. And so when we see a population like this in the height of growth in the 1st of July, this a collection of individuals near Ponder, Texas, which is in a road that I swear maybe 3 people to 10 people on a weekly basis pass by it. But this ended up on iNaturalist. Anyway, in this population, what we would do in September, October, is to actually paw through each of these individuals, count the number of leaves produced by every rootstock, and then those that had flowers, actually pull the flowers and then count the number of seeds that are actually produced by every individual flower. And we drop the seeds right where we found the plant, so we don't actually remove the genetic potential of the sexual reproducing individuals. Now, the plant defends itself as the common name suggests, by exuding a very sticky amber-colored resin. And as you handle the plant at any time of the year, you walk away with sticky fingers. And this is one of these parasites, potentially, in a moth that has as yet been not identified. And it is such a mimic for the bracts that actually are found to subtend the capitula on these lateral branches of a flowering individual. So what we did is we actually then looked at survivorship and calculated some of the vital statistics that one can using these demographic data that we collected. Now, this is a comparison of 8 populations that we have characterized over the course of the years. These are all from 2019. And what they do is it's a static life table. So we're looking at log survivorship on this axis, and on the x-axis, we're looking at stage class. And I use stage class in lieu of age class because we don't know for sure that there is a rate of leaf production. And I think the fact is that it's variable enough depending on environment that it is a relative measure at best. But this is typical of what we would call a type 2 survivorship, where the average probability of mortality is equal no matter what the age of the individual. And these locations, so this is the southeasternmost individual, Rutherford Lane. Mason Mountain Wildlife Management Area is the westernmost and southwesternmost population. Tandy Hills is one of the northern, Oak Cliff, about the same, but they're both in the middle of an urban area. Cedar Hill is a nice conservation area. Stella Rowan, likewise, is a conservation area. So many of these areas that we've looked at Silphium's prosperity is in areas that are set aside for conservation-related activities. When we actually then use the static life table, and I'll show you an example in just a minute, we calculate what's called net reproductive value or reproductive value. And reproductive value is the average number of individuals produced through sexually reproducing by an average individual over their lifetime. So it's the total number of offspring that could be produced on average for an individual in that population, or you can look at it by age class or stage class. And what we see here is that to sustain itself, white rosinweed requires a net reproductive value of 1 to be maintaining, to sustain itself. And we see that in some cases, in fact, we've got quite a range of variation. And in other cases, we have no variation at all. In fact, that the populations don't seem to be effectively reproducing themselves at all. That is, the net reproductive value is right at 0. Now, there are some. Mockingbird and Midlothian seems to be doing okay. Oak Cliff is, well, somewhat holding its own. But we get out to Mason Mountain, out in the middle of the wilds of Central Texas, or in Cedar Hill, we see, in fact, that the populations are maintaining themselves. Though there is the chance, because we're looking at the 95% confidence interval here, there is a probability that these are also not effectively reproducing themselves, leaving only 2, Mockingbird Nature Park and Stella Rowan Prairie in South Fort Worth, that are actually sustaining their populations. We then look at Rmax, or the intrinsic rate of increase. And this is basically the difference between birth rates, per capita birth rates, and per capita death rates. And so a value of less than 0, it means the population is declining exponentially, and that greater than 0 is going to be increasing exponentially. And if you note here that most of the populations are, in fact, include 0, only Mockingbird and Stella Rowan that are actually effectively reproducing themselves. And this is what a static life table looks like: stage or age classes, number of individuals per age class, total number of seeds produced by individuals of that age class, the reproductive rate, the fecundity schedule, number of seeds per individual, overall individuals, and then survivorship, age class specific survivorship. And then what we do is we sum, that is survivorship times fecundity, and that gives us RO. And then we take that and we multiply it times the age class and divide that by RO, and we get what's called generation time. And what this indicates that we've got at this particular location in Stella Rowan, the average age at which seeds are produced is the 6-leaf stage. And this gives us a feel for sort of how the populations are operating. And it helps to know how the population is structured, because we see here then that the vast majority of the individuals at Stella Rowan are actually not engaged in the reproductive process, right? They're growing and developing. So there's only a few individuals that are actually engaged in the reproductive process to propagate the population in situ. So from that, we then move to genetic estimates of structure. And this was worked on by a student of mine who is just in the midst of graduating this week. And what we did is we submitted about 100 samples for genomic analysis. And what they did is we extracted the DNA, they sequenced it, and then the genomic analysis was then sorted in a manner so that we could actually characterize the structure of the populations. And we used a program known as Structure. And what this does is it seeks to see how much genetic variation can in fact be subsumed under certain categories of population similarity. And so we ran this at N of 1, or a K of 1, a K of 2, a K of 3, and K meaning the number of distinct clusters of populations, all the way through K is 10. And the optimal, that is the best solution, ended up showing at N or K equals 4. So of all the populations we looked at, and this is throughout the entire range of the species, it seems like the populations seem to cluster in sort of 4 major groups. And we can see here on the top banner here, we see that there's the Trinity River drainage, and this, as I'll indicate down below in the next one slide as well, that some students of mine back in 2019 at the Texas Plant Conservation Conference at BRIT actually hypothesized that in fact the genetic relationships of the species actually could be traced through the major watersheds that the species covers, which is to suggest that actual gene flow or ancestry and descendants actually are confined to particular regions of particular drainages. And that Robbie McManus's analysis actually seems to hold this as true as well. So we've got a number of individuals that are genetically quite distinct from anything else, and they share some of that genetic variation with some of the other populations nearby in the Brazos River drainage, the bluish color. Then the Trinity populations, mostly light blue, all seemingly sharing a great number of allelic variation here. This would be the SNPs, the single nucleotide polymorphisms on the x-axis, and the populations, the individuals themselves on the on the x-axis, and the alleles on the y-axis. So the Trinity seems to hold a very sort of general similarity of alleles that are shared only slightly with the Brazos and the Colorado. And then, so the Brazos is the sort of the most diverse. It contains both the 4 classes, the light blue, the 2 blue, and the 2 green, whereas the Colorado only slightly looks at, holds some of the genetic variation in the blue, and is mostly of the 2 major groups, the light green and the green. And if we look at that then a little bit closer and look at the sort of the sub watersheds, the same seems to hold forth. That is, the groups seem to cluster together, showing like with similarity. And so populations in these stretches of watersheds seem to hang together fairly effectively. Now, Robbie thought that there was some evidence that some of the higher elevations seem to be somewhat distinct from all of the others, but only the high elevation types as well.
So Bruce,
Yeah.
I got a couple of questions.
Shoot.
One is, I read on wildflower.org, which is the Ladybird Johnson Wildflower Center website, that white rosinweed is long-lived and its taproot reaches 15 feet. Is there a way to age a plant based on the number of leaves? And seed is available at Native American Seed and probably other sources. Is it wise to introduce this species in other locales in which it is known to occur, or where it's diminished in population?
Well, that's it. Okay, so there's lots of details in there. Yes. So when they built the Chisholm Trail Parkway, some of the local naturalists actually dug out a couple of individuals of Silphium, and the extent of the root penetration was actually greater than 15 feet. It was like, it was 50 feet. It went down and down and down and down and down and down and down. So it has the ability to really persist. And I have thought maybe what we need to do is, get down real deep and then carbon date some of the cortex, see how old it is. Introducing, so I've spent, since I first started to try and germinate these in 2008. I was unsuccessful with getting consistent germination until I actually included limestone in the pots with the seeds. And now I can get germination rates exceeding 85% consistently if I make sure that I've got enough limestone in the soil mix. Now, what that suggests to me is that sort of broadcasting or spreading the seed of Silphium is only going to be effective. That is, it's only going to pick up the opportunity to grow if, in fact, it finds that limestone, that either the basement or it's got limestone on the surface. So I'm not convinced that I bought some of the Native American seed that had Silphium in them, and I spread it on my at my lake house in Possum Kingdom. Now there the sediments are slightly different. And I watched where the Silphium went and sort of stuck, flags in the ground to see if it would germinate. And most of the time, the seed didn't work its way into the soil well enough so that it actually was capable of germinating. And about half of them were actually removed by local seed eaters, birds and rodents and the like. Now, that shouldn't dissuade anyone from trying to get it to grow anywhere, right? And so I have, like I said, I'm pretty successful now at getting it to germinate, but the conditions have to be right. I don't know if I answered that question completely. I wouldn't dissuade anybody from spreading the seed. The chances of it actually growing, though, are going to be fairly improbable unless you're going to sow them on limestone. Another question, Suzanne?
Yes. If the Silphium are mowed frequently enough, will they put more energy into leaves and then try to quickly send up a flowering stalk?
I've not seen that. I've seen them mowed in south of Abilene every year, around Stephenville and over by Glenrose, where they get out and they mow right away at the end of June when the grass gets a certain height. And once they're mowed, they don't come back and flower, in my experience.
Okay.
That whether they invest more in leaves, I tried to test this hypothesis back in the 2009, 2010, 2011, when we had a real massive drought here. And we did see a slight increase in the number of individuals that were producing leaves. So what we saw was in a relative abundance in the age stage classes that were above 4, 5, 6, 7, and 8. And there was a decline in seed production. So it suggested to me that, in fact, the drought had an impact on growth strategies. I've not seen it in association with mowing, though. Usually when you cut it off by,
Yeah.
middle of June, it's done flowering.
Yeah, it's already put all of its energy into that, and that's all it's going to do.
Till next year, and then sometimes
Yes.
it comes back. But I've seen some of those populations dwindle, and then depending on the conditions or when they mow, if the mowing regime changes, they'll come back. So mowing isn't detrimental necessarily.
Okay. So at the end, when you're finished with your presentation, I have a photograph that one of the members sent to share on my
Oh,
screen
good.
for you to take a look at. So,
Yeah.
so anyway, please continue.
Okay, well, more of the same. So the single nucleotide polymorphism data matrix, actually, when you analyze it and sort of reduce the dimensionality, it actually does show that there's nice patterns of the Brazos here in blue, the Colorado in red, and the Trinity River drainage in green, supporting the idea initiated by Alyssa Hutchinson and Ms. Lothingham, that in fact what we're seeing is sort of the populations prosper relative to one another within these watersheds. And likewise, if we look at it even closer at a slightly different degree of resolution, populations found in the same sub-watershed of the Brazos, right, and the Trinity, and then the Colorado seem to hang together, suggesting that what gene flow does exist seems to be mostly local. And we still haven't documented effectively who the pollinators of this species is. We've documented a vast number of moths and wasps and bees that seem to hang out and take pollen and or nectar from it, but whether they're effective pollinators or not has yet to be determined. Something else for us to look at in the future. So then the last little bit of this is when I first started looking at Silphium, and I looked at some of the capitula, the flowering heads, or we look at the stem here, and it's riddled with holes. And then many of the flowers actually never mature because they're infested by some sort of a parasite. And so what we did is we looked in the literature, and sure enough, there's a fairly common chalcidoid wasp that appears to parasitize Silphium. But when I collected a bunch of the capitula and some stems and brought them in from the cold and let them sit on the bench top over the months of December, January, February. In March, they hatch. And this actually is a sort of I don't know if it's going to show it or not, but this is a video, and the pupa are actually coming out. So I've had a number of students. It doesn't look like I'm going to be able to show it, but they seem to be parasitizing the disc florets of the white rosinweed capitula. And these are some of the species. And our efforts to identify who's eating who, there's various classes of chalcidoid wasps. There's the parasite, that which probably is is Antistrophus silphi, that seems to be a seed head parasite on not only Silphium laciniatum, but more than likely Silphium albiflorum. And we identify them at this point. I've sent them off, but the people at the Smithsonian have a backload. They have a long list of things that they're supposed to be doing, and I'm sure mine don't get any priority at all. That we see the Antistrophus and then the Euritomas. And it's suggested that the Eurytoma is actually parasitoid on the Antistrophus, or the Trimeris, which could be in fact a parasitoid on either Eurytoma and or Antistrophus. And as we continued to look and watch them hatch, and I'm not an entomologist, so I don't have any training, and it took me about 6 months to realize that what I need to do is I need to watch it. If it's gonna start to hatch, I need to actually look at the pupa and watch it hatch, as opposed to pulling it out of its puparium, squashing it and extracting its DNA and using barcodes. And I've used both the COX-1, the cyclooxygenase, the electron transport chain, but I've also looked at the internal transcribed spacer and the 18S and the 28S subunits of the ribosomal DNA sequences. And they're not consistent. The different barcoding sequences don't actually identify the same species every time. And so I'm at a little bit of an impasse, but what I'm waiting for is the people at the Smithsonian, they give me some identifications so that then I can use that as material for barcoding, but more importantly, to see who is eating who. Because the other class, beside the parasites and the parasitoids, are the inquilins that actually live in the same puparium and just sort of exclude the other one. In any event, the compass plant, Silphium laciniatum, it's a sister species to white rosinweed. And this is where we're headed next. So we've got the genomic samples all set, and we're sending them for sequencing, and we're going to see if in fact we can't find which of the populations or which area in the range of Silphium albiflorum is actually probably ancestral or at least basal. And so the process of evaluation of the ancestry of white rosinweed is in fact in process. So that's sort of a ragtag. I don't have much more. I can talk about seed germination. We've done some experiments. I need to actually identify some of the people who've been involved over the years. And these are ISD and educators and students of my own that have worked on this. And then one who, J. Niall Fisher, who's accompanied me on, I would say probably 75% of my forays into the field. He can identify a limestone outcrop from Google at 10,000 feet, and he can get me there in a minimum of mileage. The rest are students that have worked on this in one form or another. And then most of the support has come from the Texas Higher Education Coordinating Board, the Vivian Redlich Trust from JP Morgan, and then the Sam Taylor Fellowship from the General Board of Higher Education at the Ministry of the United Methodist Church.